is the topic of today's concern. So I'll start with sharing my slides. Welcome all of you. So this is our university. The topic which I have chosen is applications of MALDI-TOF in biological research. So first, let me brief you, because I know most of you are from biology background, brief you what is a mass spectrometry, and later switched over to principle of MALDI-TOF-MS, and then applications explaining my own work. In any biological research, whether it is basic microbiology or microbial identification or biochemistry, diagnosis of pathogens, in environmental microbiology, the analysis, biotechnology, the useful products being detected, quantified, and industrial microbiology. So in all these fields, characterization of the biomolecule is extremely important. Say you have a protein or a peptide or a carbohydrate or DNA, characterization becomes important. If it is in protein, you will know molecular weight of the protein, structure of the protein, or sequence, amino acid sequence of a particular protein, and then relate to biological activity and things like that. So in every field of biology, the biochemical characterization becomes inevitable in order to understand the process. A major concern for biologists is to develop a sensitive method, how minimum sample is required, and a reproducible method for identification of biomolecules. So you have an electrophoresis, or you have a column chromatography, you have cell detection, many other processes. The major concern is to have a sensitive method, a minimum concentration of the sample to be detected, and it should be reproducible for biomolecular identification. For this, for example, antibiotics or enzymes, various biological activity guided assays and screening procedures have been developed. For example, bioautography, the spore germination inhibition, or disc or well diffusion method for antibiotics assessment. And all these are biological activity based assays. Mass spectrometry has been used as a potentially viable physical method for characterization of biomolecules. Originally, the mass spectrometry was developed in chemistry based on physical parameters like molecular weight of or a mass of a particular compound or sample. So various mass spectrometric methods have been developed, but later it got entry into biological field and it has revolutionized our information in biology. Well, the basic scheme of mass spectrometry, if you are taking, there is a sample chamber. On your left-hand screen, you can see the sample chamber where we provide conditions for ionization of the molecules. So it should be an ion source. These ions developed from a compound or from your sample will be allowed to move through a time of flight tube where ion separation based on charge and mass ratio. So the smallest molecule will move faster, the largest molecule may move slower. And by this, in the time of flight tube, you have ions getting separated. And these separated ions will go to mass detector where it will be expressed as a mass spectrum. So on x-axis, you have molecular weight. On y-axis, you have relative intensity. So whatever the molecules you allow for the detector, molecular weight is being displayed as a spectrum on a computer screen, where later you will be able to get a printout and analyze and things like that. So basically, in a mass spectrometer, you are ionizing the sample ions getting separated based on their MZ ratio, and the mass detector will identify molecular weight or mass of each biomolecule. As far as time of flight tube is concerned, though it is being referred as MZ ratio, there are options for only allowing positive ions or only allowing negative ions. So in such cases, we will directly get the molecular weight, as I will show you in the later slides. What is an ideal method for characterization of required? Like minimal sample preparation. If you say 1 kg of the fungal mass you require, or 1 kg of a tissue is required for analysis, then it is not good method. So minimal sample preparation, or if you have 10-15 steps for purification or analyzing the sample, then it is not a good method. So minimal sample preparation should be there. It should permit automatic analysis. For example, if you have 1,000 samples and if you have a lot of preparation time for samples, then it is not a good method. An ideal method should allow automatic analysis of series of biological samples, whether it is plasma or urine or tissue extracts or directly the cells, like that. Then it should also provide adequate information about stable characteristics of the organism. For example, proteins, cellular proteins, extracellular proteins or secretory proteins. They provide the physiology of the cell. So if you are interested in protein characterization, it should be in a position to provide stable character, the protein consistently present in the cell or released into the medium. So one such method is MALDI-TOF-MS. What is MALDI-TOF-MS? The acronym matrix assisted. Matrix assisted. Matrix is a compound, a small molecule or mixture which will help assist the sample to get ionized when the laser energy is provided. So matrix assisted laser desorption and ionization. When you mix the matrix with the sample, matrix will absorb the laser energy, transfer it to the sample molecules, make the sample molecules ionized. These ionized molecules will fly off from the sample chamber. They move in time of flight tube, as I explained earlier, based on their MZ ratio, and then go to the detector. So you have matrix assisted laser desorption ionization. Time of flight mass spectrometry is one of the techniques available for biological sample analysis. The major goal of MALDI-TOF is to vaporize and ionize the analytes. Analyte is your sample without fragmenting. Most of the other mass spectrometry earlier than this were heating the sample, destroying the sample, denaturing the sample. This is referred as a soft ionization, where without disturbing the or fragmenting the molecules, you will be able to get the mass detected. So by incorporating the analyte into an excess of matrix, as I mentioned, matrix is a chemical which absorbs laser energy, transfer it to sample molecules to make it ionized. So sample preparation. What are the samples in biology which you can use? Intact cell, bacterial cell, a nerve cell, human nerve cell has been analyzed by MALDI. Tissue samples directly, virus particles, cell lysates, microbial extracts, mycelial extracts, or secretory compounds from the medium. Purified proteins can also be analyzed in less than 5 minutes. One of the important requirement here, advantage is a short time required for analysis. Otherwise, for molecular weight determination of a protein, you need to run an SDS page and then identify using the marker the relative molecular weight of the sample, which at least takes 6 to 8 hours. Sample preparation, then purification if required, then separated by electrophoresis, SDS page, and then determine the molecular weight. Whereas here, crude samples being put in, you will be able to detect within 5 minutes. So one of the ideal character required is a rapid method. Then it is very interesting to note diverse samples like air. If I were in a food industry or a pharmaceutical industry, people would like to know whether any microorganisms are there, any other compounds are there in the air, indoor air. So the air can be sucked directly by an air sampler, and the air sample can be placed on MALDI-TOF for the detection. Then soil extracts, foodborne pathogens being detected, physiological fluids, clinical specimens like plasma, sera, or urine, the sputum can be processed and analyzed. Aerosols containing bacterial and fungal spores from a pharmaceutical industry or a food industry can be analyzed directly, trapped and analyzed. So having said that, how this works, there are different methods. I have a instrument, how it works. I have developed based on the principle of MALDI-TOF animation in order to show you. In the next slide, it will come. Before that, just have a look at how the sample preparation. The sample solution has to be prepared. You can take extracts of the cell or directly cell suspension to be placed on a sample plate. Like a slide, this in the microscopy, this one is a stainless steel plate having the capacity to hold 2 microliter of the sample. 2 microliter of the sample. That means the amount of sample required here is very, very less. And hence it is a highly sensitive method. And first you put the sample and allow it to air dry. This is called dry droplet method. A mixture of matrix and the sample can be added directly and dried. This is also called seed layer method. So, or you put one layer of matrix, another layer of sample, another on the top, another layer of matrix. So 2 layer methods you can follow. Sorry. As I mentioned earlier, matrix solution used are alpha cyano 4 hydroxycinnamic acid, CHCl-CA, alpha cyano, cinnaphinic acid, gensic acid, ferulic acid. These are the compounds which easily gets ionized under laser energy. And then not only they get ionized, they will also transfer the energy to excite the electrons from the sample and make them ionized. So any ionizable molecule like protein or peptide are present in the sample gets ionized. If the molecule or the biomolecule or any compound doesn't have the capacity to ionize, then you cannot use MALDI-TOF-MS for analysis. So this is the sample plate I was telling. You can see there the small circles on the stainless steel plate you have, which holds 2 microliter of maximum sample. So 2 microliter of a suspension of the sample with the matrix to be loaded. Either you can have 1 microliter of the matrix, 1 microliter of the sample, like that. Different permutation combination you need to use based on the samples you have, and sample matrix ratio becomes one of the important parameter in ionizing. Then as many in this single plate, as many as 400 samples can be loaded. So automated system is accessible for you to process large number of samples routinely. With replicates, you can process at a time 200 samples. This is the overall MALDI mass spectrometer of Bruker Daltonics present in proteomics facility, molecular biophysics unit, Indian Institute of Science, where I worked for my samples to be analyzed. The left-hand corner, you have a box-like structure here, which holds the plate and gets closed. The laser falls onto the machine, and this is the time of flight. It goes to the detector and then displayed on your screen. The vertical MALDI mass spectra are also available. Overall, the scheme is same. You have an ion chamber, you have time of flight tube, the electromagnetic field which allows the ions to move, and a detector. How exactly this occurs? These are the 2 slides. This slide and the next slide is extremely important, which I have developed myself with after understanding the principle of MALDI-TOF-MS. The sample plate, the stainless steel plate, will hold the sample mixed with the matrix. S indicates the sample, M indicates the matrix. This becomes an analyte. Then there is a laser energy, nitrogen laser. The laser beam comes and hits. You can adjust the intensity of the laser beam also in order to make ionization possible. Once the laser beam comes and hits, matrix readily absorbs the energy. transfer it to the sample molecules, and they get ionized. You know, positive ion, negative ions, depending on the composition, they get ionized. Some are non-ionizable molecules, and they get dissolved from the sample plate, enter into the time of flight tube. So ionizable molecules, unionizable sample components are left behind in the ion chamber itself. So now you have assembly of all the ionized molecules on the left-hand screen, that is ion chamber. When you put time of flight tube into positive ion mode, that means it allows only positive ions to pass through. The electromagnetic field in the time of flight tube provided in the instrument will allow only the positive ions to move. You can also put the instrument on negative ion mode to characterize molecules which are ionized, having negative charge. So when you allow this to happen, the smallest molecules move faster, followed by other. Then based on their mass, they get separated. All positively ioned molecules have moved into the detector through time of flight tube, where they get separated. So time of flight tube essentially separates the molecule based on MZ ratio. Here you have allowed only positive ion to separate it. So based on their molecular weight, they got separated. The negatively charged ions and non-ions ionic molecules are left behind in the ion chamber itself. This is the basic principle of MALDI-TOF-MS. So if you look into it, the overall, there is a sample chamber, the laser beam comes and hits, the molecules gets ionized, they get separated there based on their molecular weight and goes to the detector. When we see in the detector, we will get to know what are the molecules associated with the sample. Here, I am presenting a part of research which we have carried out. In particular, Indian Academy of Sciences provided me an opportunity to go and work in Indian Institute of Science with Professor P. Balaram. And there, the facility was there. This organism, referred as trichoderma, one of the important antagonistic organism being used as biological control agent to antagonize other plant pathogenic fungi. It's a biocontrol agent, and now commercially also available. There are a lot of mechanisms by which trichoderma suppresses plant pathogenic fungi. For example, it is highly, this is 3 days old plate. Within 3 days, it covers 9 centimeter plate. That means fast growing, highly competitive to colonize the soil, and then produces hydrolytic enzymes to degrade pathogenic organisms cell wall, then produces several antibiotic compounds to kill other fungi. So a potentially biocontrol agent is here, which we have analyzed through MALDI-TOF for the first time. I had several isolates obtained from different places, and the pure cultures are maintained in the slants, as you can see on your right hand side. One of the group of compounds produced by trichoderma are referred as peptibols. Peptibols, they are peptides containing AAB, alpha amino isobutyric acid, which is a non-standard amino acid. Non-standard amino acid means it is not present in the protein. It gets synthesized by non-ribosomal synthesis. Peptibol synthetase enzyme is involved in synthesizing these peptides. Residues, and the C terminal is ending with the terminal is having alcohol. Carboxy terminal is ending with alcohol. They are antibiotic peptides produced from trichoderma and related fungi. The interesting parameter is that there is a lot of microheterogeneity having these amino acids, and there are 330 peptibols have already been identified, characterized. And one of the basic requirement for peptibol is it contains high number of AIB, alpha amino isobutyric acid, the amino acid. The C terminus is the alcohol moiety, as I mentioned earlier. For example, phenylalanine, anilinol, leucinol, isoleucinol, like that. The last amino acid is converted into alcohol derivative. They are microheterogeneous polypeptides. I did mention From trichoderma only, 330 peptobols have been identified, which differ in their molecular mass by 14 Dalton. Instead of one amino acid, the other amino acid is inserted. The overall sequence is same. A one amino acid is changed, which results in 14 Dalton or 16 Dalton differences. For example, instead of AAB in one peptobol, alanine is present. So it differs from 14 Dalton. AAB to valine, leucine to valine, glycine to alanine, glutamic acid. Like this, the interchanging of amino acids is the basis for microheterogeneity, molecular weight differences of 14 or 16 Daltons. So this is the basic background which I started working. In fact, I went to characterize molecules from trichoderma and got the MALDI-TOF instrument there. Out of curiosity, I just processed my sample and we got wonderful results. So this is the mass spectrum, MALDI-TOF mass spectrum obtained for a trichoderma. As you can see here, the x-axis contains MZ ratio, but all these are positive ion charged molecules, and hence it directly reflects on molecular weight. The y-axis is relative intensity in terms of percentage. You can see this cluster of molecules obtained. Please remember, I have used here directly the spores of the fungus, mycelium of the fungus. No sample preparation, only directly the spores are kept on the MALDI sample plate with the matrix alpha cyano and obtained these peaks. As you can see here, 1714 is one maximum peak here, and the next peak is 1757. That means 16 Dalton difference. 1727, 1741, 14 Dalton difference. This is group of peptobol obtained having 17 residue. 17 amino acids are there in these peptobols. The other isolate had two groups of peptobols, one 17 residue containing peptobols, the other one is 19 amino acid containing peptobols. These are excellent spectra they were giving, and Professor Balram was excited to see this spectrum, and we continue to do many other, so many microheterogeneity group, even a single amino acid variant, we are getting different mass spectra. As you can see here, 1790 and 1804, that means 14 Dalton different. 1918, 1932, 16 Dalton, 14 Dalton difference. Like that, each one of the peptobols, in spite of having single amino acid variation, they were reflecting in the molecular spectrum obtained, mass spectrum obtained. So the other strain of trichoderma had 11 residue or 12 residue, 14 residue and 17 residue. We had strains, 32 strains of trichoderma being processed. We had 17 residue peptobol producing trichoderma group, 14 residue and 17 residue peptobol producing group. This is 12, 14 and 17 residue peptobol producing isolate. The other one is 14 and 17. That means chemical diversity in terms of peptobol was possible to be detected at the cell level itself. That's why this is called cellular peptidomics. Cellular peptidomics, analysis of peptides in the cell. And interestingly, I got a paper in 2001, Liu et al., 2001, single cell MALDI. The laser beam comes and hits only 2 micrometer region of the sample plate, 2 micrometer region. That means a single bacterial cell, a single fungal spore would be sufficient to produce such beautiful spectra. What does it mean? You can analyze a single cell and the molecules which are getting ionized. So when I look back, Birkbeck College, London, Department of Crystallography, they are maintaining peptobol database. Birkbeck College, London. Peptobol database is peptobols, peptides. They are synthesized non-ribosomal synthesis, and hence they are not available in the protein database. So the crystallography group maintains this sequence of characterized peptobols. For example, 11 residue peptobols produced by Trichoderma herzianum, one species, Trichoderma herzianum, 55 different peptobols have been identified, characterized, sequenced. Look at this trichoresin. It has got 11 11 amino acids and molecular weight is 1160. Herzianin 1161. You have trichoresin 2 1174. 14 Dalton difference for the first one and third one. The second one and third one 14 Dalton difference again. So such is the micro heterogeneity available. You can see here the single molecule was a single amino acids being varied in the particular position, resulting in differences in the molecular weight and which can be detected using MALDI-TOF. So 14 residue peptobols in a similar manner, then 18 residue peptobol, 19 residue peptobols. So this is the data already available in the data bank database of peptobol database by Birkbeck College, London. What I did, we had different strains of trichoderma. Earlier ours was Kuvempu University, so we have given Kuvempu University microbiology culture collection number. The calculated molecular weight based on the amino acid sequence and observed molecular weight. So when we have only observed molecular weight as an experimental proof, we have a theoretical value to be obtained by analyzing the trichoderma. The nearest one is able to identify the molecule already present. Within 2 minutes, directly from the cells using MALDI mass spectrum, we were able to identify the peptobols present in different isolates of trichoderma. So you have trichoresin. So we did not characterize this, but directly based on the mass spectrum, we were able to know this compound is being produced by this isolate. So all 11, 14, 18, 19 residue peptobols were identified. So this got published in from Fungal Diversity Research Series as well as in a Frontiers in Fungal Ecology. The other interesting molecule when we were processing this in the small molecular range within 2000 molecular weight. The higher level, we were able to get another protein, hydrophobins. Hydrophobins are the proteins secreted by some of the aquatic fungi and they get assembled on the hyphal surface, making the hyphal surface hyphae to float on the water surface. It has got an hydrophobic region, hydrophilic region. Hydrophilic region is attached to the mycelial fragment hyphae. Then hydrophobic region is outside, which makes the aquatic fungal cells to float on the surface. So when they are released secreted by the hyphae, they are monomeric and then get assembled on the surface of the hyphae. Interestingly, we got a peak at around 7,000 plus range. And we were contemplating what is this protein, whether it is a protein or not. So we did an interesting experiment of labeling the nitrogen present in the protein. But how do? I use N15 labeled ammonium nitrate into the medium. Zepecdox medium we took and grew. Normal Zepecdox medium will have got ammonium nitrate, N14 labeled. N14, it is normal. N15 is having one nitrogen being incorporated, one atomic weight gets increased, and hence as many nitrogen is getting incorporated into the peptide, you will have more as many molecular weight differences. For example, in the lower of the B indicates normal ammonium nitrate being used, N14 labeled, the molecular weight is 7,383. Interestingly, in the same isolate grown in a N15 labeled ammonium nitrate, we were able to get 7,467. So there was molecular weight shift for the same, indicating that this is a protein. And when we looked into the literature, this protein corresponds to hydrophobins. We sent this for a publication in Microbiology journal. All the teachers here, you should know this, an interesting part. We sent this and the referee had asked many questions about the assumption of this is a hydrophobin protein. And the paper was not accepted immediately. So to answer those questions, we were pushed to work little more, characterizing this hydrophobin protein. By another 2 months, a article got published in the same journal by so-called the referee. They got the idea of doing MALDI-TOF-MS for characterizing hydrophobins, and they did. They had used 2 additional controls for the same methodology. What are those additional controls? A purified protein, hydrophobin protein, they put on the MALDI-TOF, they got the same peak, indicating that it is hydrophobin. The second, hydrophobin minus mutant of trichoderma was placed. This peak was absent, indicating it is hydrophobin. By using 2 controls, additional controls than ours, they could get the paper. We lost the race. This was in 2006. So biochemical characterization of trichoderma strains in terms of diversity and hydrophobin protein variation. I did mention we got trichoderma isolates having 11 residue or 12 residue, 14 residue, combination of 14 and 17, like that, indicating metabolic diversity. Cellular level detection, it is extremely important. Without any pre-processing of the sample directly from the cell, it was possible to detect chemical diversity in terms of peptobols. The other interesting organism we took is Aspergillus terreus. These are the 6 different isolates of strains of Aspergillus terreus capable of producing lovastatin anti-cholesterol drug. So this we had initially seen for antifungal compound. We found interesting differences in the isolates as far as antifungal compound production is concerned. As you can see, morphologically also in a cultural character, they differ. So we made a native page of mycelial proteins. We got several protein differences between, for example, if you see the second lane, the one which I have marked with an arrow mark is absent in the other. So regular electrophoresis for mycelial protein is one of the methods for distinguishing strains and species of fungi. It has been done already in many cases. When we put these for mycelial cellular type of spectra, as you can see here, one molecular weight of 12,494, that peak less in the other strain. So we were able to see 3 such peaks which have been marked there were absent in the other strain. What does it mean? A cellular extract was able to provide in MALDI within 2 minutes the differences between strain differences between the organisms. Likewise, we got all the 6 strains characterized. One of the alternatives for MALDI-TOF is the LCMS, liquid chromatography mass spectra. Though electrospray ionization, the sample in the form of liquid is sprayed in minute droplets and which facilitates ionization, and then it gets detected. We had my Ravindra Babu, who got recently his PhD. So what we did, we made use of weight of the compounds, and the spectrum obtained was uploaded into a progenesis software for identification of the compounds. This is one such retention time is showing in the lower graph, and the molecular weight of different peaks obtained is showing in the upper. In case of Aspergillus nodulans, one is ginipin, molecular weight of 227, which corresponds to blue pigment. Another is diazine, yellow pigment, 255 is the. We were able to identify tetrahydrocurcum near the yellow pigment. molecular weight of 373. Then a reddish brown pigment juglon was identified in Telleromyces fumicolosus, molecular weight of 175. The software associated with the mass spectrum will identify the compound already characterized. So you will know whether your organism is having this molecule or not. Then they have now extended MALDI-TOF for various characterization of microorganisms. For example, IMTech Chandigarh, the MTCC, Microbial Type Culture Collection, they have a MALDI-TOF based identification services. American Type Culture Collection, ATCC, has got a microbial culture collection. Even if you have a mixed sample, you process it on MALDI, get the spectra, and the spectra are compared with the known spectra to identify the organism. One of the company the instrument with which I work, Bruker Daltonics from Germany, they have produced a software called Biotyper. In Biotyper software, particularly for clinically important microorganisms, they have got 4,000 spectra already available in the software. If you have isolated a bacterium, take the MALDI mass spectra, submit the spectrum to the software Biotyper, it will give you the identity of the organism. The other one, Anagnos Tech company from Germany, they have developed a special software, spectral archiving and microbial identification system, which will provide the spectra to be analyzed and gives you the report of identification of the organism. So overall, how to use this Biotyper? You have a culture, pick up a colony, plate directly onto the MALDI plate, get the spectra and compare the data with the identified spectra, and you will get to know which is the organism. So the whole process takes only 5 minutes to finish off. Particularly in case of clinical samples, this becomes highly beneficial tool for diagnosis. Identifying molds, multicellular fungi it is a complex aspect of microbiology, but having cultural condition, colony morphology, and so many things. But Biotyper, a novel liquid array, has made it possible for filamentous fungal identification also, without doing colony morphology, waiting for 7 days to grow and all that, with the reference library available. If the reference library is not available, then you need to identify the organism, create the reference library. So what are the current applications of MALDI? One, rapid. I did mention within 5 minutes, you can identify the molecule present. It is highly sensitive because only 2 microliter of the sample is enough. The process is very simple. Then sample preparation is less than 5 minutes. Per sample analysis time is about 5 minutes. Identification of strain, species, genus, specific biomarkers from direct cell analysis is possible. Continuous monitoring of the air in the workplace, like a food and pharmaceutical industry, is possible. Microbiological investigation of water, medical diagnosis, chemotaxonomic classification, detection of proteins, peptides, nucleic acids, phospholipids, and other metabolites. One limitation is that non-ionizable compounds cannot be detected. Even analysis of fungal metabolites like mycotoxins, aflatoxins have been done. Cyanobacterial metabolites have been done like microcysteines. Direct peptide profiling, PMF, peptide mass fingerprinting, where you can increase the laser intensity, make the molecule fragmented, you determine the molecular weight and determine the amino acid sequence. Whether all this is true, the applications, I have here the literature support to do that. For example, this is MALDI-TOF-MS being used for 123 filamentous fungi, where they were successful in 114 filamentous fungi identified. So success rate of identification was almost 92%, 93%. Then in another case, for research diagnostic purpose, you have to identify the fungus and then do up to DNA analysis and things like that. But in MALDI-TOF, directly you identify the organism. Mass spectrometry based proteomics detecting plant pathogens. Extracellular expression of recombinant protein in Ganoderma was identified using mass spectra. Characterization of proteome of an apple responsible as a host protein expressed when a fungal pathogen enters has been identified. This has been published in Journal of Proteomics. Then you have positive blood cultures, serum, antifungal compounds. First, all these are identified using MALDI-TOF-MS in a medical mycology laboratory. The implementation of MALDI-TOFs in medical microbiology laboratory has revolutionized practices significantly. Turnaround time, the amount of time required for processing the sample has been reduced for identifying yeast and filamentous fungi. Significant advances have been done for accurate species identification and strain identification. Then purification and characterization of antifungal protein from a plant bitter gourd. This is published in Protein Expression and Purification. A panel of 290 molds and yeast were identified, obtained from veterinary samples like pets, cattle, and animals. So for veterinary microbiology also, this has been highly useful, much earlier than the DNA sequencing methods. So identification achieved at the 89% success rate. Then food samples were analyzed for identifying the organism with almost 95% of the success rate. So MALDI mass spectra of Aspergillus flavus for aflatoxin production. Non-producing strain and producing strain, you cannot distinguish morphologically. For aflatoxin detection, you need to do TLC or HPLC and then go for toxicity analysis. Here with the MALDI-TOF-MS, two peaks, one is 304, another is 383 molecular weight, were identified for distinguishing non-producing strain from producing strain of Aspergillus flavus for aflatoxin detection. Then MALDI-TOF-MS based on foodborne yeast, casein, milk protein, protease, metal protease from Thermotomyces. Then identification of fungi from indoor environments, directly air being sampled and 72%. Comparative immunology, the development of antimicrobial peptides in frogs have been identified. That's why I did give my title as biology. So whether it is a plant protein, whether it is an animal protein or a peptide is possible to be identified. Microbial species, biodiversity, settling dust in a pig forums. Veterinary microbiology, yes, it is possible. In environment comparison very short time. Only the major limitation is the instrument costs 3 crore rupees. But the instrument present in Institute of Science is a national facility. One can approach there for your sample analysis. So in conclusion, characterization of biomolecules is possible with the MALDI-TOF-MS. Only the requirement is the molecule should be ionized. Non-ionizable molecules, for example, lipids, cannot be easily characterized using MALDI-TOF. Microbial strain specific molecules, as I mentioned, peptobols in case of trichoderma, hydrophobins in case of trichoderma, was possible for identification. And in medical mycology, yeast, medical bacteriology, many E. coli, Salmonella, Shigella have been identified. So diagnostically important. Biomolecular profiles. Chemotaxonomy is one important aspect in polyphasic taxonomy. Identification of a microorganism, a fungus or a bacterium, or looking at its biomolecular profiles in order to assess intraspecific level based on protein or peptide was possible. And animals is been possible as give many references. So using MALDI-TOF-MS, it must be possible for us to analyze the samples without much time or extraction process is required. You can use various present in the MALDI-TOF sample. If there are any questions, I will be happy to answer. Thank you very much.
Sir, good morning, sir.
Good morning.
Sir, myself, Dr. Prabhu, sir, from the Biotechnology Department, Davangere University, sir.
Okay. Please.
So this is very wonderful presentation, sir. Really inspired us, sir.
Hello.
Hello, sir.
Please check or put it in the chat box. Hello.
Sir, good morning, sir.
Good morning.
Sir, it was very wonderful, sir, and inspired us a lot, sir, by your presentation, sir.
Thank you.
Sir. Sir, I have one question, sir.
Yeah, please.
Sir, recently we are isolated one yeast isolate, sir.
Yeast.
Yeast, sir. That we want to identify. Hello.
Yeah.
That we want to identify through MALDI-TOF, sir.
Yes.
We have sent the sample for that MALDI-TOF characterization, sir.
Okay.
Okay, sir.
Where?
Further, sir, to the that's what, sir, MTCC, sir, MTCC, Chandigarh.
Okay, MTech. Yeah.
MTech, sir. Yes, sir.
Right.
Sir. Further, we are not for identification. We are in confusion, sir. Actually, biotyper identification. So, how it will be helpful to identification that yeast isolate, sir?
See, both Saramis and Biotyper
Yes, sir.
software already have known organisms spectra, mass spectra.
Yes, sir.
For example, Salmonella or Saccharomyces or E. coli, like that. Known spectra are available. Whatever the mass spectra you have got, you need to submit for comparison. So the most of the molecules which are common in your organism and already known organism, then it will give you the identity.
Yes, sir. Definitely, sir.
Yeah.
Yes, sir. Thank you,
That's
sir.
how you do traditionally, identification. You look at the characteristics of an organism. For example, if colony parameters, biochemical tests you do, and compare it with the known always using a dichotomous. Here, instead of dichotomous key, it will come to mass spectra already present in the software of different organisms.
Yes, sir.
Okay.
Yes, sir. Thank you, sir.
Yeah, please.
Yes. If any other questions, otherwise I want to give vote of thanks to the sir.
Always actually participant, though it is online, You need to be active, you know.
Hello, sir.
Ah,
So myself is Venkatesh, sir.
yeah, Dr. Venkatesh, yeah,
I
please.
have one doubt, sir. So
Yeah.
after MALDI-TOF, we'll get a MBZ value. So by using the MBZ value, if you are blasting in mascot search engine, so mascot search engine based on their MBZ value, only they will identify the similar peptide. Sir.
Correct.
So how much confidence should be there for identification, sir? Confidence.
As far as identification is concerned, the consistently see whatever the software what they have developed, biotyper, E. coli, for example, E. coli, let us say 15 peaks are there.
Yes, sir.
15 peaks are there. That means you are able to identify 15 different masses, right?
Yes, sir.
So like that, they have consistently got present in only in E. coli, not in other organisms.
Yes,
so
sir.
when you compare your mass spectra, it will be able to give 100% identification. I showed one research article where 123 organisms they used. They were able to get 114 consistently same. So
Okay,
you can also do all traditional method of identification and
yes, sir.
get a confirmation with the MALDI-TOF also in
Okay.
your research.
Sir, for a protein identification, sir. So organism identification, okay, but protein identification conventionally we can't. Very difficult to conventional method. But when we based on their MBZ value only, we have to identify approximately 50%,
Protein.
70%.
See, 1 minute. MALDI-TOF will give you only molecular weight of a protein.
Yes, sir. Right,
It
sir.
will not give any other character of the protein.
Yes, sir.
If you are looking at the enzyme activity, you need to analyze its enzyme activity.
Yes, sir.
If you are looking for any other toxicity or biological activity, you need to do for all other characterizations.
Yes, sir.
Directly, only molecular weight is given. It will not identify the protein. Further characterization, if you see, one of the parameters for protein characterization is determination of molecular.
Yes, sir.
Instead of SDS page, you can do it in 5 minutes here.
Yes, sir. Yes, sir. Yes, sir.
So now most. But all other protein characterization you need to do.
Yes, sir. Okay, sir. Thank you, sir.
Sir, good morning, sir. I am
Any
Divyendu
other?
Shekhar Mohanty. Yes, sir.
Yeah. Dr. Shekhar Mohanty,
I have
please.
a question, sir. Yes, sir. Sir, what is the cost for the purified protein MALDI-TOF, sir? What are the average costs of the protein to be to determine the molecular weights through MALDI-TOF?
Many companies are which do per sample 250 rupees.
Yes, sir.
Up to 2000 rupees. Institute of Science charges 2000 rupees. There are companies which can give you for 250 rupees also per sample.
Sir, when the protein sample is not pure or not homogeneous, then what type of problem we are getting?
You will get many peaks of protein mixture present there. Then you need to know which one protein you are looking for. For example, I did show you there were many other peaks, but 7300 was corresponding to hydrophobins. So when you have a crude mixture, you will get as many peaks as many proteins present. Then you need to look for which protein you are interested in.
Thank you, sir.
Sir, this MALDI-TOF comes with the database search also?
Database search is accessory. In the Institute of Science, they work on proteins though, so they don't have microbial identification system. But it is an accessory one. MTCC has got MALDI-TOF with the data software.
Which
So
is
depending on for example, if you are interested in microbial identification, then you need a software. Just to know the like I did in my Peptobol case, only the peaks were available. I compared with the already known database, which is freely available in the Birkbeck College, London website. So I did not use any other database.
Okay, okay, sir. Thank you.
Sir.
Okay.
I'll present vote of thanks, sir, if no other questions.
Thank you, HRDC. 1 minute before you present, I would like to thank HRDC of University of Mysore and Professor Kumar for providing me an opportunity. All the participants, wish you all the best. Thank you very much.
Thank you, sir.
Sir, good morning, sir. Thank you for giving me this opportunity to present vote of thanks. I am immensely thankful to Professor S. Shishupala, sir, Department of Microbiology, Davangere City, Davangere, on behalf of HRDC 16th Refresher Course, University of Mysore, for sir's own ideas of expertise in your research field and elegance of explanation of everything, sir. Once again, on behalf of HRDC and me, we must need to mention our deepest sense of appreciation. And I also extend thanks to all the participants for their cooperation and patience hearing. Thank you, sir, once again, and thank you one and all. Thank you, sir.